Thank you for coming back.
Thank you for having me. And thank you all so much for coming. And I should say that I was so excited to do this last spring, but also very excited to come back because this is a group that I feel I guess an alumnus of. So I started my career at Stanford with a Stanford graduate fellowship, and that was one of the reasons that I came. So when they asked me to do this, I was like, yes, totally, of course. So I am here today to talk to you about speaking science. And I'm assuming that at this point in your careers, you've all given talks before. So I'm not going to go into basics today. I'm not going to tell you that you should use certain fonts or certain background colors, although I am happy to talk about those things if you're interested. I wanted to try and talk today at a little bit of a higher level, to talk about ways that we can engage and keep an audience engaged in our research, which is hard, right? We work on really specialized, technical, difficult things, and explaining those things to an audience can be challenging. So the plan for today is that I'm going to spend about 35, 40 minutes speaking, maybe longer if you guys have great questions, and I hope you have lots of questions and that you should feel free to interrupt me. And then I'm going to turn it over to you. So a major part of this is for you to get a chance to practice delivering your research. So hopefully you're all sitting next to someone that you've never met before, because if you're not, I'm going to make you move. So don't get too comfy. So at the end of my talk, I'm going to give you some time to think about how you might tell somebody about your research in about 2 minutes. So you encounter somebody on a plane or in an elevator. How do you get them interested in your research? What do you tell them? And then you'll practice telling it to each other and give each other some feedback. And then we will turn you loose on the food. So all of these things are required before you get whatever it is in the back that smells delicious. So again, I want to reiterate, please do feel free to stop me anytime. I want this workshop to be as helpful for you as it possibly can be. So if you have questions, comments, disagreements, anything, please do feel free to stop me anytime. So I want to start out with a quick poll. I'm curious, where are my biologists? All right. Engineers? Many mathematicians in here? Physicists? Nice. Did I miss anybody? Awesome. How many of you have a parent or a sibling or a grandparent or an aunt or an uncle, significant other, that does not work in your field? So all of us, at some point or other, have to be able to talk about our science at the most basic levels, right? And we also have to be able to talk about it at the highest levels to colleagues in our lab. And the techniques that I want to talk about today should bridge all of those types of conversations. And just for the record, these are my non-scientist parents. They're both social workers. They have no idea what I was doing here. None. But I'm working on it. They know it has something to do with Schwann cells. So that's our mission today. How do we get people, whether they're our parents who have no idea what we're talking about, or colleagues in our field, to understand and engage with our science? And in my mind, this involves 2 steps. First, you have to bring them in. You have to get them engaged. You have to get them to listen to you in the first place. And the easiest way to do that, actually, is just with how you say things and the body language that you use. So we're going to talk about some really quick, easy things that may seem obvious, but not everybody uses, to help you engage an audience right off the bat. And then once you have their attention, you can keep their attention, both through how you structure your content, so how you present your information, and in the visual aids that you choose. So those are the 3 areas we're going to cover today. So let's get started with some quick tips for how we can bring an audience in right from the get-go. First, please prepare yourself. Think about what it is that you might want to say. Because at some point, you will be sitting next to someone at the Thanksgiving table and they're going to ask you what you're working on. You're going to meet a colleague at a poster session. You will have to talk about your research at some point. So it's worth just taking a little bit of time to think about it. And once you've thought about it and figured out what you want to say, practice it. And don't just practice it a little bit, practice it a lot. Because the more comfortable you are with talking about your science, the more confident you're going to come across to the audience, the more prepared you will seem. And once you've done that, once you're ready to actually engage with these people, remember to welcome them. And this is where body language really comes in. It takes you less than 1/10 of a second to look at someone's face and decide whether or not you like them, whether or not you trust them, which is really terrifying when you think about politics. So the audience is reading information from you right away. So you want to control what message your body is sending. So take a step forward and open your posture out to greet the audience. And there's a big difference between a posture like this, which is welcoming and confident, and a posture like this, especially if you're hiding behind a podium or trying to get into the corner of the room, right? This says, I don't want to be here. And we've all seen, okay, some of you can't see me, but we've all seen people standing next to a poster like this. And you never visit those posters, right? So open body language, eye contact, smile, and engage with the audience. And please remember to be excited. I know a lot of you are in your first quarter here at Stanford, so you haven't had time yet to become jaded. I promise it will come. But we all get to that point in our research where we're just exhausted. And even though we are so excited to be here at Stanford, somewhere in the back of our brains, it doesn't necessarily show. So remember why it is that you came here. Remember why it is you are dedicating your lives and your evenings and your weekends to pursuit of knowledge. And let that excitement show. And if you're not excited, if you're at that point in your PhD, it's usually like around year 3, where you're really not excited, lie. Because if you're not excited, your audience will not be excited. But also remember to please be yourself. When we get up in front of a room to present, we tend to put on our fancy presenter hat and our fancy presenter shoes and become someone else entirely. And we become overly formal, and we start using big fancy vocabulary to try and impress people. It's a conversation. There's just a lot of you, and you're not really talking back at me very much. But it's still a conversation, so you should do all the things that you do in a normal conversation. Things like making eye contact, smiling, cracking jokes if you're that kind of person, using gestures. So be yourself. And also remember to use inclusive language, and I mean that in a couple of ways. When we write in the sciences, we use passive voice. The experiment showed. The results indicated. When you're giving a presentation, I want you to let the word I into your vocabulary. I found, I discovered, I showed, we found. And when you're referring to the audience, refer to them as you. We're having a conversation. We're real people. We're not disembodied third-person voices in the room. The other thing I mean is to avoid jargon and abbreviations. You may know exactly what a Schwann cell is. Anybody know what a Schwann cell is? It's a small percentage. And everybody in your lab may know exactly what a Schwann cell is. But most of the people in this room have no idea what a Schwann cell is. And that does not mean that the people in this room aren't smart. You are all brilliant. So I should be using language that everybody in the room can understand. And if I have to use a word like Schwann cell, I should explain to you what it means. It's a really fun cell that hugs. I also mean that you should avoid abbreviations like the plague. So in my PhD lab, we use the abbreviation SC all the time. And within our lab, it could be Schwann cell or it could be spinal cord. So we would confuse ourselves within our own group. So again, just like jargon is familiar to you, abbreviations may be, but it's not to anybody else. And even if you tell yourself, okay, I explain what it means, you're asking a lot of the audience to expect them to remember what that abbreviation is going to stand for. Now, I recognize you don't always want to say the whole thing. Amyotrophic lateral sclerosis sucks as a phrase and as a disease. So if you have to use the abbreviation, make sure you remind the audience a couple times what it stands for, or put it up on the slide to make sure that they follow you and they know what you're talking about. You can also use analogies, metaphors, real-world examples to help bring things to life for the audience, to help relate them to something that they already understand. So for example, I know that my biologists are a minority in this room. So most of you probably don't think much about cell adhesion. But cell adhesion is how all the cells in your body stick together, and it's really important because otherwise you'd be a blob on the floor. And as it turns out, there's lots of ways that cells can do this. So one way is called an adherens junction, and the cells will extend little processes like Velcro and stick to each other. And that is in contrast to something called a tight junction, which is where two cells will get right up close to one another, and those proteins are going to stitch between the two cells like quilting. Now you may have never heard of an adherence junction or a tight junction, and you may hope to never hear of them again. But I'm assuming most of you have encountered Velcro, and most of you have a quilt on your bed at home. So by using ideas that you're familiar with, it gives you a way to latch into the idea and to relate it to something you already understand. So that's one of my challenges for you today. When you're thinking about how you might talk about your research to a stranger, try to come up with some sort of a metaphor or analogy to let that person in. And please, beware the curse of knowledge. I'm not just telling you this because we're close to Halloween. The curse of knowledge is the idea that once you know something, it's really hard to remember what it's like not to know it. We get so trapped in our own specialized tiny worlds that we forget that they're specialized tiny worlds. So I would encourage you to have one person that you use as your test, as your filter. So when I'm talking to a group that is not my lab group, I would ask myself, would my mother understand this? And if the answer is no, I need to rethink how I'm going to talk about it. So try and step out of yourself and your specialized knowledge before you talk to other people. And finally, I would encourage you to ask questions. So in the simplest sense, you can ask questions like, do you understand? Are you following? The audience will appreciate if you check in with them and give them that chance to give you the feedback. But I also mean that when you ask a question, you're inviting the audience in. So even if you're not asking a direct question like, do you know someone who's affected by multiple sclerosis? You can ask a rhetorical question or frame your ideas as questions. Use a research question instead of a statement. And that invites the audience to think about it and allows it to become more of a conversation. So I hope that some of these seemingly straightforward techniques will work for you when you're trying to bridge that gap right off the bat, to bring somebody in. And then my hope is, of course, that nobody ever looks at you like Beaker is looking at us now. So are there any questions about this section before I transition to the next part of my talk? So once you've got their attention and you've welcomed them in and started this conversation, how do you keep them involved? Because in some senses, that intro is the easy part, right? It's the science that's clearest. It's the introduction. You haven't gotten into the nitty-gritty yet. And if you're going to jump in a little further, now you might be getting into more complicated information, more complicated terms and ideas. So how do you keep that engagement? We're going to start by thinking about how you do it with content. How do you structure your information to make it accessible? So I like to use what I call the bow tie method. It's very scientific. And it just means that in the beginning, you start broad. You start at the point where everybody will understand. You give context. You tell me what it all means. And then in the introduction of your talk, you're going to zoom in a little bit. You're going to start to give me that detailed information. But then at the end of your talk, you're going to blossom back out, put everything back into context, and give me some sort of a conclusion. And it doesn't matter to me if your talk is 2 minutes or 2 hours. This structure tends to work. So let's break down those individual elements a little bit more. The introduction of your talk has to do a couple of really important things. First, it helps to let people know where they're going. Robert Sapolsky on campus has done lots of experiments with rats that show that they are way less stressed about getting an electric shock if they know it's coming. People like to be prepared. So tell them what they're going to hear about. Give them some sort of a roadmap. This is especially important if you're giving a longer talk, because you want people to be able to scaffold that information and know what's coming. You should also provide any context and background information that they might need to understand your talk. And here, knowing who your audience is key. Because the context and background that you might need to give is going to vary depending on who you're talking to. So do a little bit of research to figure out who that person is so you can adapt accordingly. And finally, engage your audience again. Why should they be listening to you? Why should they care about your content? And you can do this by creating relevance, by telling stories, or maybe by creating a sense of mystery. It's worth remembering, too, that your passion and your excitement is often enough of a reason to listen. If you're so excited and you can't wait to tell me about this, then I probably can't wait to listen. So let's think about these in a little bit more detail. So one way to provide relevance is to use big, scary statistics. 1 in 1,000 people are affected by muscular dystrophy. That's a big, scary number. That means that probably nobody in this room has muscular dystrophy, right? But if you were to compare that to a statistic like 1 out of 2 people over the age of 85 is going to have some form of dementia, now you're looking at your neighbor going, well, it's me or you. That's a much more immediate piece of information. People are terrible at dealing with big numbers, even if they're engineers, when they're listening to them. So use numbers that are manageable and easy to understand. Don't say 49%, say half. Numbers are good, scary statistics are good, but stories are better. There was research that was done that was shown if you're trying to get somebody to donate, they are more likely to donate if you tell them a story about one individual that's emotional versus if you tell them a terrifying statistic. So this is what geneticists use to show a family tree. And this is the family tree of a family that I worked with when I was doing my postdoc here at Stanford. And this was a family, they came into the Stanford clinic with 2 sick little girls. And they had no idea what was wrong. And we were part of the research team that worked together to figure out what was making these girls sick. It's a sad story, right? But now you all want to know. I've told you a story and I've created a sense of mystery. You have to stick with me to find out how we figured out what was wrong with these girls. So I grabbed your attention. Now I have to get to the tricky stuff. Now I have to get into the body of my talk. And this, I think, is the most terrifying part. Because this is where all the complicated information is. All of your hardcore technical data that can be hard to talk about and hard to visualize. And if you present it the way that it's shown here as one giant stream of information, you will lose everybody in the room. I promise. And we all know that this is true, right? If you're listening to a presentation and it's a constant barrage of facts that aren't linked together, that aren't put into context, we wander off. They're asking too much of us. Help your audience out. Give your bow tie teeth. So what do I mean by this? I mean that you can dive in to get really specific with your information, but then you should come back out and tell me how it fits into the big picture. Explain to me what it means. Then you can set it up to dive back in and out and in. And that helps me follow with you and gives me these chances when you surface back out for me to catch my breath and make sure I understand what's going on. And by the way, I'm happy to send the slides if that would be helpful. But it's not enough just to give it teeth. You have to link the teeth together. They have to make sense as a narrative. You have to tell a story with your data. And one really nice way to do this, which fits very well with how we do science, is to ask questions. They keep coming up. So you have a hypothesis, you do an experiment, it leads to another question. So you come up with a hypothesis, you do the experiment, it leads to another question. That's one really simple way to link your ideas together. So now you've come to the end. And I beg of you to not make the most common conclusion mistake that I've seen. It comes in 2 forms. And in either case, it's the sign of an unprepared person. There's phenotype 1, which is the person who didn't plan their time appropriately and got to the end of their presentation and realized that they still had 10 slides and they had 30 seconds left. And that's the person that frantically flips through all of their slides talking really fast. And then there's the opposite of that, which is the person who didn't plan a conclusion at all and got to their last data slide and said, okay, yeah, any questions? Which I think is an enormous wasted opportunity. You want the last things the audience hears to be the most important things. We know as audience members that it is impossible to pay attention for an entire talk. All of you will wander at some point today. I am not offended. I know it will happen. I know it's not my fault. It's hard to pay attention. So chances are somebody is going to miss one of your lovely fins. So at the end, take that opportunity to summarize. Remind them about what they might have missed. Cue for them what those most important things are. You should also put your information back into context. Now that you've added this new piece of knowledge to the world, what does it mean? What are the next directions? What are you going to do tomorrow when you go back to lab? And finally, re-engage that audience. Remind them why this is so important to them. And that circular structure actually helps people remember. It helps them pay attention. You may have noticed that there's this interesting parallel structure, right? In the beginning, this is so cheesy sounding, but it's true. You tell them where they're going, and at the end, you tell them where they've been. On both sides, you want to give them context. You want to give them some background. And on both sides, you want to engage with the audience and tell them why it's important. So perhaps my bow tie isn't so much a bow tie, but rather a diamond ring. So I hope that this is helpful for you when you think about structuring your information the next time you have to give a talk. Are there any questions about content structure before I transition to my favorite part, which is visual aids? All right. You guys just want the cookies. I'm on to you. It's cool. So, as I said, this is my favorite part. I love visual aids. I love PowerPoint. I think that when you're talking about science, it's almost impossible to give a presentation without some sort of visual aid. Because science is complicated, and we're not that good at describing things with only our words. And I don't know about you, but I cannot draw. So PowerPoint is a savior for me. Having said that, though, it's worth thinking about whether PowerPoint or Keynote or Prezi or whatever LaTeX, whatever software you want to use, is the way to go. Because sometimes a whiteboard or a chalkboard is the best solution. I think if you're, for example, working through a series of equations, that could be really hard with PowerPoint, but could be incredibly powerful on a whiteboard. And it's actually harder to give a talk with a whiteboard. So if you, for example, are trying to impress somebody and you have mad whiteboard skills, that's a way to do it. Because giving a good whiteboard talk is hard. But I love visual aids. And I love them, one, because I'm a visual learner. And so for me, they help me pay attention. They help me understand. And I know that there are other visual learners out there. But two, research has showed us that if we match a visual cue with a verbal cue, people are more likely to remember. The problem is, if there's a mismatch between those two cues, if your slides don't match exactly what's coming out of your mouth, you'll distract people, and they'll be less likely to remember. So hitting that sweet spot is incredibly important. So because I love PowerPoint so much, and I know these things to be true, I have come up with 3 guiding principles that I use for every talk that I give. And because it's my talk, I get to be lofty in my title. So the first, perhaps unsurprisingly, is audience first. And that means that every single thing that you put on your slides is for your audience. Your slides are not your notes. They're not cue cards for you. And I recognize that that is asking a lot of you. It's asking you to be much more prepared and much more aware of your material because you don't have that cue up on the slide. You can, however, have the cue in notes or in presenter view on your screen if you feel like you need that backup. But everything that's up on the slide should be for your audience. And that often translates into having less stuff up on the slide is more effective. And it's worth asking yourself too, is everything that's on my slides that's for my audience appropriate for them? Are they going to understand it? And we'll get to that more in a minute. Second, I would encourage you to be creative. Every time you open up PowerPoint, it gives you boxes to fill in. It is just as easy to delete those boxes as it is to fill them in. So just making the choice to have a picture or to avoid a bulleted list is enough to make your presentation stand out because it's a little bit different. And you will have more fun. And finally, I must ask you to consider aesthetics. And I mean that in 2 ways. First, your slides should be a very happy, friendly place for the audience to rest their eyes upon. That means no terrifying color combinations. That means no teeny tiny font. That means no horrifying, overwhelming slides. It also means that in every room there is somebody like me. And that person is totally anal retentive, obsessive compulsive, insane. And if you have one word misspelled, one thing misaligned, one random dot that Excel put there and we don't know how to get rid of it, that is all that I will see. And I will be incredibly distracted for the rest of your presentation because I'm watching for the next time that it happens. And I promise you, there is one of me in every room. And I would hate for that one crazy person to be your advisor, or the person that you're asking for a job, or the person you're trying to get funding from. So take that extra 10 minutes to really polish your slides. Put them up on a big screen if you can. Get a friend to look at them, because I know We all hit that point where we're not even seeing them anymore. Take that time to make them look really polished and professional. So having said that, let's look at an example that I made so we can totally make fun of it that fails catastrophically on all 3 counts. It hurt my soul to make this. Right? Anybody understand what's going on? Awesome, another success. Right? What does this slide tell you? The person who made this is lazy. They don't care about their audience. They don't care if the audience knows what's going on. Many of you look both transfixed and perplexed, which is not really what I want out of my visuals. And you may be saying like, yeah, okay, you made this up to look totally awful. The biggest thing to happen in physics recently was the discovery of the Higgs boson. And this is a representative example of what the slides look like in the presentation that was used to announce to the world the biggest thing that's happened in physics for a long time. Complete with, by the way, Comic Sans. So I'm not making this up. Slides like, and you've all seen slides like this, right? That are just terrifying, that don't take the audience into account at all. And it's worth noting that the coverage of these slides included things like this. So this is in German, but I think we can all guess what this says. And just in case you can't. So visual aids matter. People pay attention to them, and they get distracted by them, and they will focus on the Comic Sans and not on your amazing data. So visual aids matter. So how do we deal with that? How do we deal with the fact that we have such complicated data to share and not a lot of time most of the time, right? I know you're all busy. So how do we deal with our data? How do we face these complicated things? So I have another set of straightforward things that hopefully will help you accomplish this task. First, remember to beware the curse of knowledge. Just like with your words, you need to take a step back and ask yourself of your visuals, will the person I'm talking to understand this? Is there a way for them to get in there? So knowing who your audience is, again, is key. I can't emphasize that enough. So for example, when I talked about my own research, instead of starting with that terrible collage of black and white and gray, I would start with something like this. Because you may not have any idea what a Schwann cell is, and you probably don't care, but everybody knows what a fish is. And I can move from here to a point of complexity, because you're all smart. I just need to start from the place where everybody understands. So for example, with the Higgs boson, instead of those horrifying pea green slides, they could have started with something like this, which when I saw this on the web, I was like, this is the coolest image I've ever seen. I have no idea what it is, but it's the coolest image I've ever seen. This is something that is relatively simple, catches your eye, and gets you interested and engaged. I did look this up. This is a proton-proton center, and there's photons coming off. Don't ask me anything else. But notice what I just did there. It's not enough to just put something like this up. You have to explain it, and you have to physically interact with it. So point at things on your slides. Guide the audience to what they should be looking at. And I want to have a brief aside about this thing. I highly encourage all of you to get one. They're like $30. It's called a clicker or a remote. And it does have this useful feature of allowing me to control my presentation without being tied to my computer. And that means that you can interact with the audience more and you can interact with your slides more. However, it does come with this really dangerous thing. It comes with a laser. And there were a lot of engineers in this room, so we know lasers are dangerous. And they're especially dangerous in presentations because it's really easy to badly use a laser pointer. First off, laser pointers are excellent indicators of how nervous you are or how much coffee you've had that day. They're almost impossible to hold still. Versus, for example, your finger, which if I put it here, I know it will stay here. I trust it. The other thing about laser pointers is that because I'm right-handed, if I wanted to use this laser pointer to point at something on the slide, I have to turn my back on you, which means I'm turning my voice away from you and I'm turning my back on you, which is rude. You want to face your audience as much as you can. So again, if I use my hands, I can use either arm, and I don't have to look away from you. However, sometimes we have no choice. Sometimes we're in a huge lecture room and we have to use a laser pointer to indicate things. So again, I would encourage you practice with it so you know how to use it. And please don't do this. You're not allowed to read things and underline them one at a time. You're not allowed to spastically circle things. Once is plenty. And please remember to remove your finger from the laser pointer button before you turn back to face the room. We've all been strafed by laser fire before. Don't be that person. It's a pet peeve. I'm sorry, it's my talk. I was allowed to make that aside, but please handle with care. So back to this. It really is cool, right, though? Another trick that you can use to help guide the audience is to annotate your images. We all know when we put together a figure for a paper that you include all sorts of helpful things to help the audience understand your data. So here is that horrible gray stuff again, and I've thrown the book at it, right? I have different colors. I've pseudo-colored my images. There's labels, there's arrows, there's boxes, there's cartoons. I'm not saying you need to do all of that. And I'm certainly not saying that this is acceptable as a slide, because this is still super overwhelming, and I can see half of you trying to figure it out. We're going to talk about information flow in a minute. But try and use some of these techniques to clarify your information and help guide the audience to what they should be looking at. There is one enormous problem with this slide. Any guesses? Is anybody in here colorblind? About 10% of the male population is colorblind to some extent. And of those people, some people cannot tell the difference between red and green, which means that what I've done here is spend hours pseudo-coloring my images so that to some people, they still look gray. Right? If you are truly colorblind, red-green colorblind, these both just look gray. And as it turns out, my PhD advisor was colorblind, is colorblind, he's still alive. And he didn't tell me that fact until after I'd spent the hours putting together these figures. So if you do have to have contrasting colors next to each other, go for things like blue and yellow or purple and green, which are color combinations that everybody can see. Because there are red-green color combination colorblind people out there. And as scientists, especially biologists, we suck at the red-green color combo. It's everywhere. So the other problem with this, as I said, is that it's still just way too much stuff. I would encourage you to control information flow. Remember that you are also a source of information. The audience is also getting lots of information from you, and they should be getting most of their information from you. It's a presentation, not a paper. So you can simplify what's up on the screen, and you can control what the audience sees when. So now when I was researching the Higgs boson so I could show the example of those slides, I felt that I should probably understand what the Higgs boson was if I was going to make fun of the scientists who found it. It was only fair. So I came across this cartoon, which I think at first glance actually looks pretty good, right? But the more you look at it, the more you realize that, one, all of you are looking at it and not paying attention to me, and two, the text is really tiny and there's a lot of it. So we could apply those two principles. We could reduce what's up there, and we could simplify it, and we can break it down. So instead of presenting it in that original form, I would walk you through it something like this. This is the standard model of physics. Molecules are made of atoms. Atoms are made of a core and electrons. The core is made of protons and neutrons, and this, by the way, is about where biologists stop. Protons are made of quarks and gluons. How does it all stick together as it's flying around the universe at the speed of light? Interesting question. Well, as physicists, there's really only one way to answer that. Let's smash them together. And it turns out when we smash them together, we discover something really interesting. We discover that what's holding them together is the Higgs boson, which is the maple syrup of the universe. It kind of coagulates things and slows them down enough to stick together. So think about what just happened there. I walked you through it, I broke it down, and I used a lovely maple syrup analogy. So I'd encourage you to approach your information in the same way. Even if you're giving a journal club paper and you're just pulling figures from a paper, you can still break up the individual sections and walk the audience through it. I would also encourage you to use visual displays of data, like the cartoon that I just showed you, or like graphs. Because graphs are much clearer than tables. You can look at a graph and see the trends immediately, right? It's very clear where the relationship started to decline. However, just because graphs are better than tables doesn't mean you're allowed to do something like this. And stop trying to figure it out. It's all graphs with the same information. So if you have to show a bunch of graphs on one table, remember to try and simplify and to bring things in one at a time. So this is horrible. This is a little bit better. It's still not great, but at least you're not just bombarding with the audience with information all at once. You're giving them a chance to slowly build up to it. However, under no circumstances are you allowed to be the person that does this, which is to put something like this up and say, I know it's a lot of information. Don't worry. I only want you to pay attention to this. Why are you showing me everything else? And now I don't trust you. Now I want to look at everything else because I want to know why you don't want me to look at it. All of this stuff can be in backup slides. Let them ask you. They'll be impressed at how prepared you are. Only show them what they need to see. And when you do that, it gives you that ability to break things down even more and to really walk the audience through it. So there's this really fun feature now because Microsoft Excel and PowerPoint are linked that lets you animate graphs really easily. So you could, for example, have just the axes show up and explain to me what I'm going to see, and then have the data come in one piece at a time. And I should qualify that normally I hate animation like that, but I like it here because it gives you that sensation of length. It's also telling you information. It's not just a flashy trick. You can also think about using cartoons as visual analogies. So we know that metaphors and analogies help us understand. The same is true of visuals. So I'm assuming that this is still totally incomprehensible. Excellent. How about now? You may still have no idea what's in these pictures, but you at least know that there's something that's in the skin that's moving through something else and ending up in the muscle, which, by the way, is 5 years of research in 3 sentences. That is my PhD right there. But how much simpler and easier is it for you to understand this? And now that you get the concept, I can explain what these complicated images mean. Here's another example that I made. Super simple. It took maybe 20 minutes in PowerPoint. This is a cell that we're looking at and the nucleus, so another component of the cell. And then there's different things in the cell. You stress the cell, it's terrible. Stuff moves. More stuff moves. It's very bad. Right? Super complicated biological process made accessible through some really simple cartoons and animation. Now, I know all of you are saying, that's lovely. I have stuff to do. Fortunately, Google is here to help you out. There is probably a cartoon already out there that covers a lot of what it is that you're going to talk about. So for example, anytime I had to give a journal club paper, this is where I would start. I would do a Google image search on the topic that I was going to present on and find the best image that explained it. And that's actually my favorite way to find review articles, because the best review articles often have the nicest images. And now you have a cartoon that you can use, properly sourced. I would also encourage you to create visual transitions, and we're almost there, I promise. So there's been a lot going on visually, but I hope that there's one slide that looked familiar, right? This with a stick figure. It came back a couple of times. That's because when you think about audience attention over the timeline of a talk, right, if I were to graphic for you, so introduction, body, and conclusion on the x-axis and audience attention on the y, could you all please mime for me with your hands what you think this graph might look like? Okay. 2 general phenotypes I'm seeing here. There's the swoopers, and there's the asymptotically approaching 0. The swoopers have it, right? You come in, you have a great hook, you get into the body of your talk, all the important stuff, you lose people, we know this. You get to the conclusion, you remind them about what they missed. But by structuring your content the way that I talked about earlier, hopefully your graph will look more like this. But you can also get these peaks in attention through visual aids. Through catchy visuals that grab the audience's attention and bring them back in, and through transition visuals. So the simplest example of it is something like this. super boring, super standard, but serves a purpose because it orients the audience and lets them know that something is happening. And you can even, highlight where you are in that talk. A more complicated version might look something like this, where you start with a simple model in the beginning, you build things through each of the 3 parts of your talk, and at the end you have a lovely conclusion picture to show them that's now familiar and they've built towards and they understand. So if you put this on the talk, it would look something like this. So I would really encourage you to use a device like this. Sue McConnell calls it, she's a professor in biology, she calls it the home slide. It's an orienting place for the audience. It's a familiar place for them to get back into your material. For a 10-minute talk, maybe you don't need it. But for an hour-long lecture, it's hugely helpful. All right. Where's my mouse? Sorry, I'm trying to subtly do a trick where I skip some slides and failing. I would encourage all of you to use presenter view, because instead of clicking maniacally through the slides that I don't want to talk about, I'm going to very gracefully just skip through them and remind you that you should think outside the slide. So think about embedding videos in your slides, embedding sound clips to help bring your slides to life. Think about using the whiteboard. Think about using a prop or a demonstration as part of your presentation to really bring an idea home. So I hope that you can use some of these tools the next time you're faced with giving a talk, whether it's to your grandmother at Thanksgiving or at your next scientific conference. We talked about using your body to bring the audience in and your voice to engage them right off the bat. And then we talked about keeping an audience engaged, both by how you structure your content and in the visual aids that you choose. So I really do hope that the next time you're faced with your parents or whoever it is that's not in your field, you'll be more excited and more ready and more willing to tell them about the amazing work that you're doing here as part of your PhD. I just want to quickly show you some of the resources that I used in putting together this talk, and as I said, I will send the slides, so don't worry about frantically writing stuff down. And I very much encourage you to come check us out in the oral communication program. We are housed in Building 250 on the main quad. There are graduate student tutors that you can meet with throughout the year. They are amazing and very well trained and sitting in this room. And they are happy to help you with any time you open your mouth. So conference talks, lab meeting, journal club, qualifying exams, thesis defenses, yay! Job talks, wedding proposals, anything where you have to speak, we are there to help you out, and we want to help you out. So I would really encourage you to come check us out and make an appointment. You are also more than welcome to email me. I'm happy to work with any of you on your presentations or to answer any questions that didn't come up today or you don't have time to ask me.